So today topic is bioinformatics. So we were what is bioinformatics? When we look at the brief introduction of this, the field of science in which biology, computer science, and information technology merge to form a single discipline. So bioinformatics is last 10 years, 1 decade, it's emerging as a very important information technology in the field of molecular biology. So we can understand how molecular biology or life science research is advancing so fast just because of bioinformatics. So today let us discuss how bioinformatics helps to understand molecular biology in a better way. So as we look at the basic information of biology hidden in a gene that's in the form of ATGC. So central dogma principle, this DNA or a gene express and forms the mRNA. This mRNA transcript translates into a protein. So this is the overall feature or vision of molecular biology, where scientists from all over the world working how this system is working, gene to DNA to mRNA, mRNA to protein. So as we all know, the basics of our architect or structure of an organism is that it's made up of all the proteins, which are functional unit of our system, physiological system. So without protein, no physiological functions. And most of the diseases are also associated with the system. So if anything goes wrong in this flow of information from gene to mRNA, mRNA to protein, so that's the where we need to understand, where we need to correct the mistake happened. So this is trying to understand, but the huge data is hidden in this flow of information from DNA to mRNA, mRNA to protein. So in the 21st century, we look at the entire genome. So earlier we were working on single gene. So, but now we are not able to understand so many things from a single gene, individual genes. So we are looking at the entire genome. Then transcriptome. So whatever the genes express at a particular point of time, then it forms the transcriptome. Though we are trying to understand the entire transcriptome, it's like entire mRNA profile. Then comes to proteome. Proteome means the entire set of a protein which are expressed or formed at a given point of time or at the disease state. So this is how the idea came, the bioinformatics emerged from this because a lot of information. When we look at this graph, after Human Genome Project completed in the 2002, from the project onwards, there are a lot of information or a lot of data is generating from this molecular biology. This data we need to store in different databases, and the stored data we need to analyze. To analyze, we need help of a lot of bioinformatics tools. So how to understand tools means there are algorithms, or you can understand this, they are softwares. Because this is such a huge data, so when you just look at this from 2002, after Human Genome Project, from 2025, the future also predicted this billions of nucleotides, 3 into 10 to the power of 9. Human genome length is 3 billion base pairs, just one genome. So after human genome project, there are many organisms has been sequenced and huge data has been stored in all these databases. Especially in bioinformatics, there are three important databases. One is NCBI, EBI, and DDBJ. And when you come to the protein, PDB, protein data bank, this all the protein structures are stored. So like that, we see each database is specialized for that. So if you want to understand the bioinformatics, we have to go through all these websites, how data is stored, how to access this data, and how to analyze this data, and how to interpret the data. So this all is bioinformatics. So high throughput technologies. As molecular biology research is advanced day by day, so on the same speed, the lot of data is generating. I would like to mention some of the technologies which a lot of data is coming out from the next generation DNA sequencing. That means whole genome sequencing. Suppose recently the coronavirus, there are many variants we have came across. Each variant has we have to sequence to understand what are the mutations happening. So that they named it as different strains of corona. So there's a lot of data generating from next generation sequencing. And metagenomics. This is another technique which a lot of data is emerging out, that is sequencing DNA from the environment. Because in environment, only 1% of the microbes we can culture in the lab. There are so many microbes existing in the environment which we cannot grow in our laboratory conditions, but we want to understand that those microbes also. So we are using the bioinformatics tools and we are trying to understand those microbes which not able to culture in the lab. So this is bioinformatics. Next, microbiome analysis. Sequencing of microbial communities. Just now I mentioned there are so many RNA viruses, DNA viruses, and plant infected viruses, many microbes. So all these we need to sequence it, and the sequence is stored in databases, and that is also we need to understand. That is data is generating. Another data is from the microarray, that is RNA expression analysis or gene expression analysis. So by using these microarrays, we can understand the so many genes expression at a time. That is also called RNA sequencing or microarray or DNA chips. The chip sequence or clip sequence, this is all protein interaction, protein DNA interactions, because there are many proteins which binds with the DNA. For instance, in transcription, there are many transcription proteins which binds with the DNA. And protein RNA interactions, there are many proteins binds with the RNA also. So all these data which is emerging from this microarray, chip sequence, clip sequence, and finally mass spectrophotometer. This is last 2 decades. This is one of the revolution happened. The lot of analysis, or we are trying to understand at the protein level, protein expression. Earlier, we had very limited technologies to understand the protein expressions, but through mass spectrophotometry, now the n number of proteins or expression and function and the structure we are able to understand. So overall here, the lot of data is generating. So that is, we need to store in the databases and analyze. So again, building models for what's list, that is sequencing, gene knockout, microarrays, and genetic interactions, that in protein-protein interactions, protein DNA interactions, subcellular localizations, microarrays, proteomics, metabolomics. These are different, you can say, the techniques which huge data is generating. All these data we need to understand. That's the bioinformatics. Next, computational tools need to still to pathway interest from large molecular interactions databases. So it's like a huge data is in flux. So here you look at this diagram that's yellow, green, and red. These are the different gene expressions showing in different colors on a DNA chip. This data is generating, and so many structures, chemical structures of protein and the DNA. So this is how data is coming. That data, we are using the tools. Here you can see this is a tool. By using bioinformatics tools, we are trying to analyze this data and interpret what is their structure, function, and how they related to each other. What do we do in bioinformatics? Analyze and interpret the various types of biological. What we do, we analyze the data and then interpret the data. That is called secondary data. One is genome sequences. We do sequence analysis by using many tools, and transcriptomic sequences, that is RNA sequences, proteomic sequences, that is amino acid sequences, protein structures, that is the 3D structure of the proteins, and RNA structures with the loops. This is, we analyze and interpret. This type of data we analyze and interpret. For this, we develop new algorithms and tools to access the biological information. Suppose you want to access some data from any database of bioinformatics, that we require certain tools. Handle large data sets. So example, multiple sequence alignment. This is one of the tool. By using this, we can handle large data sets. At a time, we can compare and identify the similarities between the different sets of sequences or different organism sequences, and find out a relationship between data sources. And also we can find out the relation that is called evolutionary relationship, or we can develop the evolutionary tree structure. It's a graphical presentation. Just by looking at the tree, so we can understand which organisms are close by molecular level. What if all these will we learn in this course? Pairwise and multiple alignment. So in bioinformatics, we compare pairwise, mean two sequences at a time, and multiple sequences, just now I mentioned. And database search, NCBI, DDBJ, EBI, PDB, Tiger Genomic Research Institute. So there are many databases we can search, and we can take the data and we can analyze the data. And protein alignments, that means either structure wise also we can alignment and find out the similarity, and sequence wise. And DNA sequencing, so that also we can learn, that is next generation sequencing. And gene expression, that is cluster analysis, that is by using DNA chips. Phylogenetic analysis, we can do by multiple sequence alignment. And motif search. Motif means this is a functional subunit of any protein. Each protein having the many motifs, so we can understand the function. Structural bioinformatics, this is RNA and proteins. So most of the time we need to understand the protein structure. Why? Because there is a relation between structure and the function. If you want to understand the function, we need to understand the structure. That's why they use X-ray crystallography, NMR, cryo-electron. These are the three different major techniques we use to get the structure of a protein. Biological networks. So we can link each other of a function and we can understand the networks. Many different applications of bioinformatics that is basic sciences and medical applications, agriculture applications. The main goal of the basic science is understand the living cell. So far, what we understood, the human cell, according to some experts, is just 0.001%. There's a lot more we need to understand. So even after revealing the entire DNA sequence of a cell, still so much we need to understand. So for that, bioinformatics is which helps us to understand this faster. Find the function of new proteins. So it's either way you can say. We have to understand the function of a gene. So approximately there are 25,000 to 30,000 genes are there in human genome. Scientists across world, they're trying to understand the function of each gene and the protein level also. Which gene is going to code for which protein. So, and find the proteins that are unique to human. So because most of our research is focused on human research, because there are so many diseases, infectious diseases, are challenging for us to understand. We need to understand the profiles of genes and proteins. So we are in the process of trying to understand genes and proteins to solve these complex human diseases. Example, cancer. So people are trying to understand different ways, but still we are not in the able to come up with a successful medicine for cancer, tuberculosis, or many more diseases, Alzheimer's disease. Okay, I'm audible. So find the function in basic science. There are two types of research. One is basic research, one is application research. Find the function of new protein. This is required a lot of technologies, experimental technologies, under the bioinformatics helping us to understand or define the function of a protein. Discover function of new proteins. Find the proteins that are unique to human, that we can do by phylogenetic analysis. There are a lot of tools to understand the phylogenetic analysis, that is cluster W and cluster omega and MSA, multiple sequence alignment. By using these tools, we can understand the phylogenetic analysis of any gene sequences or amino acid sequences. This is very surprising for us. Comparison between the full drafts of human and chimp human genomes revealed that they differ only by 1.23%. When we look at the genome, the very less percentage similar between one genome to another genome. But the small difference, it makes huge difference at the structural level. So how can we be so similar yet so different? So this is scientists are trying to understand just 1.23% difference between one genome to another genome. It's like a mouse genome and human genome, they're very close. But when you look at the structure, there is a huge difference between the mice and the human. So humans are so complex. The system is complex, physiological system on signaling pathways, everything is different when you compare it to the mice. But still we use mice as a mice model because there are many genes which are close to human. So there are many studies says that these chimpanzees are very close to human. So, but we cannot use this as a model, but by using bioinformatics, we can understand how close we are genome-wise. But when you look at the structure-wise, this required a lot of study to understand where we are different, where we are similar. Very different and very similar, that defines the conserved sequences which share among the genomes or many organisms. So this is one of the bioinformatics data. After analyzing the genomes, this is called comparative genomics. When you compare, there is a facility or there are tools to compare different genomes and to identify similarity and the conserved, how close we are phylogenetically or evolutionary. And also we can identify what is the origin. So there are medical applications, identify the mutations that cause genetic diseases. This is a huge challenge for the scientists to understand the many genetic diseases. Till now, we don't have any defined solution or confined solution to solve or to understand these diseases. But now bioinformatics is helping us to understand how many SNPs are there in each genome. In human genome, at every 1,000 nucleotide, there is one SNP. So SNP means this is single nucleotide polymorphism, and this SNPs associated with certain genetic diseases. So at the gene level, if you want to make some therapy, or that's called a gene therapy, but it is very complicated. Genome editing is very complicated. But through bioinformatics, we are trying to understand, and also there is a possibility that we may come up with some genome editing or gene therapy, that you can identify these SNPs by pairwise sequence alignment and multiple sequence alignment, and also DNA sequencing. Just here example I'm showing, just change by one nucleotide of our genome, because nowadays scientists are talking about genome editing. Genome editing means wherever there is mutation in a genome that we need to correct. So it's not so easy. If you correct it or insert or delete any nucleotide in our genome, that may leads to changes in the other part of the DNA or genome. So that may lead to some other diseases. So here one example is sickle cell anemia. This is one of the disease. This happened due to swapping of an adenine for a thymine, A by T. So when you look at the sequence in a healthy individual, so this is A. Adenine is there in this position, in the third position. G after A is there. But when you some mutations that become thymine, A become thymine, that's amino acid become valine. So E to V. So just one change leads to sickle cell anemia. So like this, there are many mutations keep happening. I'll give you another example. Some of the cancers, so that's due to some mutations in the gene. Instance, BRCA1 and BRCA2, these are the two genes of breast cancer. So far identified, there are many mutations. Still today, we don't know which mutation is actually responsible for cancer, breast cancer. So, but all these we can understand by bioinformatics. So under medical application, diagnosis, find the features that characterize the disease states, that is gene expression. So gene expression or clustering analysis, that is done by microarray analysis. In a microanalysis, we can understand at the gene level, so which are the genes are expressing, which are the genes are not expressing. Let's say you take healthy cell and the disease cell or cancer cell. Both the cells you can express by using this gene expression methodology, the DNA chip technology. So we can understand which are the genes are expressing, which are the genes are not expressing. So further analysis we can done by bioinformatics. That's the clustering analysis. Then motif search. Motif search comes at the protein level or at the gene level. So that also you can, by using some tools, we can do motif search. So here samples, so this is the experiment methodology is mentioned here. So gene expression levels and also differences in the gene expression, that is one of the adenocarcinoma. There are 100 genes are different in the cancer tissue in different stages of the tumor were found. So they analyzed the different stages of the tumor of adenocarcinoma. They found the difference in the gene expression. So here bioinformatics helps us to understand this, how gene expression varies of development of any disease or tumor progression. Find and develop new and better drugs. So in drug designing, bioinformatics helps DNA sequencing, gene expression, structural bioinformatics, biological networks. So this is four topics, or you can say factors we need to understand to come up with a new drug. When you look at the drug designing in the pharmaceutical companies, so to start from the designing of a drug till releases into a market, it's like more than 10 years. But because of bioinformatics now, we can save 5 to 6 years. And there's a lot of money we can save, because it requires a lot of money is required to design a drug and release into the market, because you have to go through all the trials, phase 1, phase 2, phase 3, all these. Before that, 5 to 6 years, whatever, from the sequencing to identify the mutations on the protein level, structures, comparative structures. So all these we can done by bioinformatics, and we can save a lot of time and also money. Infectious diseases are still #1 cause of premature death. That's at the age of 44 years worldwide, and annually kill 13 million people. So 33% of all deaths. That's because of infectious diseases. Most of the infectious diseases we cure with the antibiotics. As the world knows that there are many antibiotics we are using, but many of them are drug resistant. That are the drug resistant again is these antibiotics. So here ribosome is the target for approximately half of the antibiotics characterized to date. So here mentioned some of the antibiotics. So if we want to understand or to change the target of all these antibiotics to ribosome, because when they develop the resistance against these antibiotics, again these pharmaceutical companies or drug designer, they modify these antibiotics against the target. Target is the ribosome, most of the antibiotics. So this is called drug simulations. So here a lot of bioinformatics involved, how to do these simulations, how to revive that drug resistant antibiotic. So here a lot of bioinformatics involved, and a lot of tools are involved. Some of the tools are publicly available to do these simulations. and also commercially also available for better understanding. So here, what exactly they do? So here there are two sites. One is bad site, one is good site. So where exactly it has to bind? So suppose if you take an antibiotic, it has to reach a target. That target, how to define the target? Earlier we were using some methodologies, but those methodologies not as accurate compared to now the technologies which we are using now, that is bioinformatics. So you can find by using bioinformatics to find a new target, because previous target is already drug resistance. So that's why we need to find out the new target. So here bioinformatics helps. So many different applications, that is personalized medicine. This is the most advanced part of bioinformatics. So here personalized medicine, this is called tailored treatments. So now, till now, the concept is one drug to all. But now the concept is changing. That's because of bioinformatics. Now, because it's the same drug, it is good for one individual. Maybe it is not good for another individual. That's because there is a change in the genome level. As we have already seen, that is because of SNPs, single nucleotide polymorphisms. These SNPs number or a location in the genome, it varies individual to individual. That's why some of the drugs are sensitive to somebody. It's not sensitive to some other individual. Now the future is going towards the personalized medicine. What the idea is that they identify the similar SNPs pattern individuals and they group together and they design the drug for them. Then that drug will be most suitable. There is no chances, there is no uh, chances of side effects or any other um, problems. Especially this is going to may help in cancer patients. So this is a new area, an advanced area. Of course, it is some of the aspects looks very broad or vague or very, it may not to fit to the current understanding of our research, but that is the future of pharmaceutical. That's called personal medicine, or this also called pharmacogenomics. So this is there are so many applications. I've just mentioned some of the applications of bioinformatics. So this is a brief introduction or applications of bioinformatics.